What if I told you that we could read history, not from books or stone tablets, but from the very building blocks of life itself, locked deep inside a 2-million-year-old tooth or a shattered bone fragment, are molecular ghosts, whispers from a world we thought was lost forever. And today, we're going to learn how to listen to them. Welcome to G Fun Facts Online, where today we're unlocking the secrets of the past with a revolutionary science called Paleo-proteomics. For a long time, if you wanted to know about an ancient creature's genetics, the holy grail was ancient DNA, or aDNA. And it's amazing. It's how we learned that Neanderthals and modern humans had kids together. But DNA is a diva. It's fragile. It breaks down easily, especially in warm, wet places. Scientists think that after about 1 million years, even in the best, coldest conditions, your chances of finding usable DNA are pretty much zero. This left huge, dark chapters in the book of life completely unreadable. But what if there was another molecule? A tougher, more resilient molecule that could survive for millions of years? Enter the protein. Proteins are the workhorses of our bodies. And one in particular, collagen, is the main ingredient in our bones. It's incredibly sturdy. And here's the magic part. The recipe for every protein is written in an organism's DNA. So by reading the protein, we can reverse engineer parts of the genetic story, even when the DNA itself is long gone. This is Paleo-proteomics, using ancient proteins as a time machine. So how do scientists actually read these ancient molecules? They use a machine that sounds like something out of Star Trek, a mass spectrometer. Think of it as an impossibly precise scale for molecules. One of the main techniques is called zooms. It's like a rapid-fire sorting system. You can take a big box of tiny, unidentifiable bone fragments from an archaeological site, zap them with zooms, and it can instantly tell you, this one's a goat, this one's a pig, this one's a deer, and wait a minute, this one is human. It helps scientists find the needle in the haystack. For a deeper look, they use something called shotgun proteomics. This is the full investigation. It doesn't just identify the species, it can sequence the proteins, revealing incredible details about evolutionary relationships. Okay, let's talk about a real-life monster quest. Meet Gigantopithecus blacki. This was a real ape, possibly 10 feet tall, weighing over 1,000 pounds, that lived in Southeast Asia until about 300,000 years ago. For decades, all we knew about it came from a few fossil jaws and teeth. Was it a giant human ancestor? A weird gorilla? Nobody knew. And because it lived in a hot, humid jungle, its DNA was toast. But then, scientists got their hands on a 1.9 million-year-old Gigantopithecus tooth. They couldn't get DNA, but they could get protein from the super hard tooth enamel. They sequenced it, compared it to living apes, and bam, mystery solved. Gigantopithecus was a close relative of the modern orangutan, their family lines having split about 12 million years ago. A question that stumped science for decades was answered, thanks to proteins. This tech is also rewriting our own human story. You've probably heard of the Denisovans, our mysterious ghost relatives. For a long time, we only knew they existed from DNA found in a single tiny finger bone from a cold cave in Siberia. But recently, a fisherman in Taiwan dredged up a fossil jawbone. It was in the wrong place, a warm, subtropical region where DNA could never survive. So scientists turned to proteins. They analyzed the jawbone's tooth and found protein variants unique to Denisovans. This was huge. It wasn't just proof that Denisovans lived far beyond Siberia, but it gave us a crucial new fossil to understand what they actually looked like and how they adapted to totally different environments. Now, this isn't easy. You can imagine museum curators aren't thrilled about scientists wanting to grind up priceless fossils. And contamination is a nightmare. Proteins from soil, bacteria, or even the researcher's own skin can ruin a sample. But the solutions are incredibly clever. To avoid destroying fossils, scientists have developed non-invasive techniques. In one brilliant study, they analyzed the plastic bags that bones had been stored in for years. Enough protein molecules had shed from the bones into the bag to identify the species without ever touching the artifact. Others use a special film to gently lift a microscopic sample off the surface. And to fight contamination, they figured out that certain chemical changes, a type of decay, only happen to proteins over vast amounts of time. So if they see that specific decay, they know the protein is authentically ancient. Paleo-proteomics is just getting started. Scientists are now hunting for the dark proteome, all the other proteins still hidden in these ancient samples. Soon, we might be able to analyze protein residues on ancient pottery to know for sure if it held milk or meat, or study fibers from clothing to see what animals they came from. We can now ask questions about the past that were pure science fiction just a few years ago. The whispers from our most ancient ancestors are finally becoming a conversation, and we're all invited to listen. Thanks for tuning in to G Fun Facts Online. Join us next time for another deep dive into the amazing world of science.